Yeah. So as you can see, this is what the basic structure of the antibody. Okay, so this is how it looks. So it's a Y-shaped structure. Okay, and it is usually having what? 4 peptide chains. So protein oriented chains, peptide chains means, okay, so they will be having 4 peptide chains, which will be H2 and L2. H2 and L2 means what? Heavy chains 2 and light chains 2. Okay, so total 4 peptide chains will be there. So what they will do is, as you can see, antigen binding sites will be there. Okay, and that antigen binding site will be having the specific protein, which is going to detect only the specific antigen. And when that antigen binds onto this site, what is going to happen is it will create a response. Okay, so whenever there is a response which is created by using this antibodies, we call it as what? Humoral. Humoral. So humoral means what? It is nothing but the response which is going to involve your antibodies. Okay, so whenever the antibodies are involved, we also call it as what? AMI. So AMI means antibody mediated immunity. Okay, so this is how the after, what do you say, the overall structure looks like of the antibodies. Okay. So now I was mentioning about the antibodies, right? So let's see one example related to this. Now, as we know, there are certain organ transplantation and stuffs taking place, right? Like for example, it may be for the heart, for the kidney, for the liver, or whatever it is, for the blood, right? So there is something which is called as what? Transplantation. So what is the relation of the transplantation with your immune response? Now, obviously, all we cannot see match anyone's kidney or liver with everyone, right? So there should be a match. Means if that particular donor, okay, is taking that particular kidney or liver from the person, a healthy person, when it is fitted into your body should not produce any antigens against it. Okay, means it should not act as an antigen. So then your body itself will be attacking your own stuff, right? So that should not happen. So hence what happens is whenever there is an organ failure, when a person requires a transplantation, okay, there you have to see whether it's accepted by that particular patient's body or if it is rejecting it. So for that purpose, what we do is you take the blood group similar, okay, and apart from that, you also have to see whether certain characteristics of the tissue are matching. So if these two things, that is your blood group as well as the tissue matches to almost or towards the 99%, if it's matching, then what will happen is you can donate that particular, or you can keep that particular liver or kidney to the patient. But here, one more thing that you have to keep in mind is to such patients, whenever you're making the switching of these organs, that particular patients must take very important thing, which is called as immunosuppressants. Immunosuppressants. So immunosuppressants means what? When you're taking an external thing and keeping it inside you, your body should not reject it. So for that, you are being given what? Immunosuppressant. So then the body will be clearly able to differentiate between the self and non-self. Otherwise, if it is not differentiating, whatever the graft you are inserting, right, that will be rejected. Okay, so this again depends upon what? Your T cells and B cells. So now here you can see this is the liver, a donor organ procurement is given. Okay, so from that, whatever the cells are there, they are coming into actions. From there, the T cells is taken. Okay, the recipient lymphoid tissue is also taken to find out the match. And then afterwards, this particular scavenger receptors are being formed. Okay, so only when you get the final cord, then only you will what transplant that particular liver into that particular patient. So we have a tissue characteristic should be matching, the blood group also should be matching, plus that particular patient should be given what immunosuppressant to prevent the rejections. Then whatever the T cells and B cells are there, they will accept that particular external organ as a self. Okay, and whatever thing you're going to insert, right, that's called as a graft. Okay, so whatever liver, kidney, whatever things are there that you're going to transplant, it's called as a graft. Okay, so this is the transplantation and immune response. Next, the active and passive immunity, as you can see over here. Now, already I told you how the structure of the antibodies and what are the roles in transplantation. Now, this is what the types, like the active and passive. Right, so in the active and passive, what is the major difference? In the active, like whenever your body is being what do you say, exposed to the antigens, it may be either dead or living. Okay, it will produce what antibodies in response to that particular antigen. But active immunity is comparatively slow because it takes time so that it could be what fully effective against the particular disease. Okay, so usually active immunity will be what in two types, like for example natural and artificial. Okay, so natural means what? Whenever you have an what do you say, unintentional exposure to something, it causes the natural active immunity. Whereas the another one active is what? Whenever you have been injected through the microorganism. For example, when you take my immunizations like vaccines, when you take, obviously it's having what microbes, right? So those are what all your artificial active immunity. That's why it's given. The first one is infection and second one is what vaccination. Whereas in the passive immunity, what is having is it's already the ready-made antibodies which are being injected into your body directly. So immediate protection it gives you, it won't take that much time as compared to your active immunity things. Right? So here also natural you have, and this is the artificial. So natural, as you can see, the mother is feeding the baby. So through the milk, what we say is a colostrum. From that, whatever the antibody is usual to the baby, useful to the baby will be transferred. And certain what you say diseases will not affect that particular baby. But on the other hand, in the artificial, what you can see is certain, like we take the tetanus shots and all those stuff, right? So those things comes under the artificial, which are also called as what monoclonal antibodies. Okay, so certain bee stings or all those stuffs are there, we take the anti venom. Okay, so whenever a baby is born, there are certain injections given to that particular baby. Okay, so those things comes under what? An artificial immunity type. Then vaccination and what? Immunization. Now, what is the difference between the two? So here in this diagram only clearly you can see, here there is an injection, here there is what? Directly consuming that particular thing. Okay, so it's this phenomenon, like for example, if I take the vaccination, this is wholly dependent upon what? The immune system memory. Okay, so depending upon that memory only this will work out. For example, this vaccines means what? It is going to contain inactivated pathogens, means that they will be leaving but it will be in inactivated form, right? So the body which is going to produce the antibodies against this antigen will already be having what? The memory cells. And what are those memory cells? As I told you, B and T. Right, so these B and T memory cells are going to recognize the pathogen quickly because you already taken the vaccination and your body has already what produced that particular memory of this particular microorganisms, and hence further you need not to worry. And what are the vaccinations which you take usually? Polio is there, then hepatitis is there, DPT vaccines which are taken for the baby, all those stuff is called as what? Vaccination. But when you come to the immunization, it is what? It is basic development of your immunity. Okay, so there is no what do you say, direct injection given which is having an inactive microbe. Okay, so in immunization also you're having like almost two types, you can say. Like for example, one is the passive, another one is what? A recombinant DNA technology. Okay, rDNA technology, which we say. So passive immunization is the tetanus or snake bites against that, whatever the ready-made things you are going to give it. That will be what? Your passive immunization. Okay, whereas in the recombinant DNA technologies, you are just going to alter that particular DNA for that particular disease. Okay, and for example, you are having the hepatitis B vaccine. Okay, so usually in market, whatever the hepatitis B is there, that is made from yeast cells. Okay, so this is what you're altering the DNA of the yeast, and then you're making this particular what? The immunization product for against the hepatitis B. Okay, so this is how actually vaccination and immunization differs. Okay, next you have is the allergies. So I'll just, yeah. So what are allergies? Okay, allergies means every human is sensitive to certain things. Okay, maybe the